Hello students, welcome to Trunkie Academy. In the previous classes we studied about the biotechnology, its principles in processes, its application in agriculture, its application in medicine, and after that we studied about the, the structure of DNA in the yesterday's class, right? Now today we'll study about the, we studied about the biomolecules, and with regard to that we studied about the amino acid structure of amino acids. Okay, so not about the biotechnology, on that was for the class 12, sorry. Okay. In class 12, for the class 12 I taught about the biotechnology and all, but for class 11 we studied with the cell structure, fundamental as a fundamental of life, and the different parts of the cells. We studied about the plasma membrane, we studied about the different cell organisms like endoplasmic reticulum, Golgi bodies or Golgi apparatus, then ribosomes, lysosomes, okay, etc. Okay, and then we started about the biomolecules, and with regard to that we studied about the amino acids in yesterday's class, and today we study about the proteins and the polysaccharides. These are the macromolecules. When we say macromolecules, it means that different monomers are joined end to end, and they have formed a large molecules. Okay, so proteins and the polysaccharides are the macromolecules. Okay, so in case of proteins, what are proteins? These are the polypeptides. Okay, and they are linear chains of amino acid linked by peptide bond. As I told you in previous classes also, amino acids are joined by peptide bonds, and these linear chains are again joined to form a large molecules. Okay, so depending on the complexity of the structure, there are different types of proteins. Okay, they classified into different groups like primary structure, a secondary structure, a tertiary and quaternary structure. Okay, so you can see here that there are different amino acids like glycine, isoleucine, valine, cysteine, glutamine, okay, and alanine, serine, leucine, aspartic acid, arginine, cysteine. So these all are joined by the peptide bonds and forming a linear polypeptide chain. Okay, so peptide bond, what is this? A peptide bond, amide is actually amide bond is a covalent bond and chemical bond formed between two amino acid molecules. Okay, so here you can see this. Okay, amino acids and amino acid, that you can see the OH group. Okay, then there is the elimination of a water molecule forming a amide bond, or you can say a peptide bond. Okay, so that how a peptide bond is formed for the formation of polypeptide chain. Here you can see this one, this is the one. Okay, the hydroxyl group, and here the hydrogen, then there is the elimination of this, and then there is the joining of this here. This joins with the there is forming a amide bond or peptide bond. Okay, so this is the peptide bond formed and then joining the two amino acid. Correct. Now we move on to each protein is a polymer of amino acids. I told you in the yesterday's also, while teaching about the amino acids, there are about 21 types of amino acids, and by permutation and combination of these amino acids only, so many different types of proteins are formed and found in the living organisms. Okay, example: alanine, cysteine, proline, tryptophan, lysine, etc. These are the different examples out of these 21 types. Okay, a protein is a heteropolymer, means, and not a homopolymer. Like in case of the some polysaccharides, okay, like in glycogen or in the starch, we find that there is a homopolymer. So only one type of monomeric unit is joined end to end and forming a homopolymer. Means, for example, only the glucose molecule, for example, the glucose joined to glucose, again joined to another glucose, the glucose is joined to another glucose. This is forming a linear chain or polymer, that will be the homopolymer. But in case of this protein, different amino acids are there. Not only like alanine is joined, or only proline is joined end to end, forming only prolines are there. No, it's not like that. So different amino acids are joined by peptide bond, forming a heteropolymer. Okay, so this is a polymer is derived from more than one type of monomer that I already told you. The homopolymer has only one type of monomeric repeating units and n number of times it will repeat, so it will form a homopolymer, like as I told you in case of the starch. Certain amino acids are essential for health and they have to be supplied through our diet. Okay, there are some kind of amino acids that is already there in our body, but there are some amino acids that are very, very essential. Why essential? Because that's not is formed or present in our body, that has to be supplied through our diet, and that plays an important role in our body. Okay, so that is the reason. Therefore, amino acids can be essential or non-essential depending on whether this has to be supplied through our diet or not. Okay, so amino acids in the human body obtained from the nutrition. Essential amino acids, you can see histidine, isoleucine, leucine, lysine, cysteine, methionine, phenylalanine, threonine, tryptophan, valine, serine, tyrosine, asparagine, and selenocysteine. Okay, these are the essential amino acids. Okay, cannot be synthesized by our body. Okay, in case of non-essential which can be synthesized by our body are the alanine, arginine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, tyrosine, asparagine, and selenocysteine. So these are the amino acids synthesized by the body, and hence these are called the non-essential amino acids. Okay, now essential amino acids, an amino acid that cannot be synthesized in our body, therefore must be supplied through diet. Okay, so must be supplied from the diet that contains the protein and these amino acids. Okay, essential amino acids with acid or base group has been shown. Here you can see this. Okay, or base group R. Okay, so here you can see the acidic amino acid with the, this is the carboxylic group, and last class I told you the acidic group or the carboxylic group, then the amino group is there. Okay, right, amino group is there, and then we have got the base. Okay, so essential amino acid asparagine and histidine, lysine, these are the amino acids with acid or base R groups. Okay, non-essential amino acids that the human body cannot synthesize for itself, and so need to be provided by our diet. Okay, so need not to be there. Okay, it's non-essential and need not to be provided by diet, because this can be synthesized by our body. Okay, so these are the, like I told you in previous slide, alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, selenocysteine, serine, taurine, and tyrosine. These are synthesized by our body. Hence, dietary proteins are source of essential amino acid. Dietary proteins, the proteins that we take in from we get it from our food. Proteins carry out many functions. Okay, they transport nutrients across the cell membrane. When I was teaching about the structure of plasma membrane or cell membrane, I told you that the plasma membrane is made of the lipid bilayer, right? And in the lipid bilayer, there are the protein channels that is passing across the forming one channel, and they are passing across the plasma membrane, and these channels helps in the transport of materials, substances across the membrane. Okay, so these proteins are involved in the transport of nutrients, then fight against infectious organisms, pathogens and germs. Like when I was discussing about the in some classes I told you the immunoglobulins, okay, the antibodies. These are proteins which are responsible for fighting against the germs and pathogens. Okay, like against the antigens. Okay, so fight against infectious organisms. Some of the proteins are hormones. Okay, peptide hormones like insulin, glucagon. These are the peptide hormones, and they are made of the proteins. Okay, with the help of amino acid bonding. So even the enzymes are also, some enzymes are proteinaceous in nature. Protein. So these are the functions of the protein. Okay, so they're involved in the fighting with the infectious organisms. some proteins are hormones, so they are acting as hormones, and some are acting as enzymes, and some are involved in the transport across the cell membrane. Now, some proteins and their functions we will discuss here. Okay, in a tabular form, you can see the collagen. Now, one of the important protein that is found in the skin, okay, and other parts. Intercellular ground substances found forming substance to give to contribute to the mass. Okay, intercellular ground substances, collagen, trypsin, enzyme. Trypsin is a protein. It is an important enzyme for the digestion of protein. Insulin, important hormone. Antibody fights against the infectious agents like virus, bacteria, and receptor molecules like sensory reception. Okay, these are also made up of protein. Okay, so GLUT4. Okay, this is one kind of important protein which is enables glucose transport in the villi, in the intestinal wall, through which the digested food material glucose is transported into the cells, and this plays an important role in glucose transport. Okay, so this is how we observe that some of the important proteins and their functions. Okay, collagen functioning as intercellular ground substance, trypsin, which is an enzyme and helps in the responsible for digesting protein. Insulin is an important hormone for controlling the blood sugar level. Antibody, or like immunoglobulin, the fight against the infectious agents like bacteria, virus, or the kind of the things they produce for the causing diseases in our body. And receptors like sensory reception, smell, taste, hormones, etc. are also made up of the protein. GLUT4 enables glucose transport into cells. Okay. The collagen is the most abundant protein in animal world, and the ribulose biphosphate carboxylase oxygenase Rubisco is the most abundant protein in the whole of the biosphere. Just imagine this. This is the important question for the general knowledge as well as for the exam. That is, the collagen is the most abundant protein in animal world. Okay, and ribulose biphosphate carboxylase oxygenase. Okay, this is a kind of enzyme. Rubisco is the most abundant protein in the whole of the biosphere that is found in both plants. So that is the most important, common, okay, and in biosphere. Now, polysaccharide. This was about the protein. Now comes the polysaccharide. Polysaccharides are another class of macromolecule. Like I told you, macromolecules means when they are formed of the monomeric units and form a large molecule, then they are called the macromolecules. So protein is a macromolecule, and we saw that these proteins are heteropolymers made up of many amino acids joined by peptide bond, and they form a large molecule. Likewise here, polysaccharide, carbohydrate. Okay, carbohydrate can be classified into three groups. That is the monosaccharides, oligosaccharide, disaccharide, oligosaccharides, and polysaccharides. Four categories rather, you can say. So polysaccharides are another macromolecules, a kind of a carbohydrate. Okay, so here we find that found in the insoluble pellet, macromolecule found in insoluble pellet. Polysaccharides are long chains of sugars, carbohydrate, and we find it may be homopolymer or heteropolymer. So in case of starch, in this diagram we can see the glucose are the monomeric unit, and they are joined by glycosidic bond. Here they joined by glycosidic bond, like we have seen in case of the protein. Amino acids are joined by peptide bonds. Here these are joined by the glycosidic bond. Glycosidic bond. Okay, but here we find that the monomeric unit is the glucose only, and each glucose are joined together. So this will be homopolymer. It's not heteropolymer. The starch is the now example cellulose is the polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. So we call that as the homopolymer. Cellulose fibrils. You can see cellulose microfibrin, right, in the plant cell. So these form the cellulose fibril, microfibrils form the fibrils, and these cellulose fibrils are then constituting the constituent part of the cell wall, providing the rigidity and hardness by the cellulose in the cell wall. Okay, there are threads literally a cotton thread containing different monosaccharides as building blocks. So these cellulose are literally a cotton thread and forming a building blocks of the cell wall. Cellulose is a homopolymer, as we have already, I have told you, all the monomers are single type. There are no two types of monomers are present. The storehouse of energy in plant tissues, yes, this is the starch. Okay, and the storehouse of energy in animals, it is glycogen. So glycogen is the, like we found the most common, the polysaccharide in plant is the this starch, but in animals it is the glycogen stored in the liver and the muscle. Okay, right. Now glycogen here, it is, this is the chemical structure of the cellular glycogen. Okay, inulin is another kind of homopolymer of fructose. Okay, that was the glucose, but here it is a made of the fructose. It's the inulin hormone. So fructose connected by the each by bonding, they are forming the inulin, a kind of a homopolymer polysaccharides in case of this. Okay, in a polysaccharide chain, say glycogen, the right end is called the reducing end, the left end is called the non-reducing end. Okay, so you can see this one. The sugar is classified as a reducing sugar if it has an open chain form when an aldehyde group or a free hemiacetal group oxidized by mild oxidizing agents. Okay, so this is the there may be depending on this, okay, open chain form with an aldehyde group or free hemiacetal group, okay, oxidized by mild oxidizing. So accordingly they are classified. Okay, so non-reducing end, sugar is not oxidized by mild oxidizing agent. Okay, so reducing type or non-reducing type depending on this. Now you can see non-reducing end is there and reducing end is there. Okay, so non-reducing end you can see here this way where it will be it's not reducing. Okay, the structure of matter depicting non-reducing end and reducing end. So this is the reducing end where we find it. Now both the cases you see the similar structure, but this will be the reducing end and this will not be reducing. Okay, just by changing the position of the atoms also it will be the reducing and non-reducing end is changed. Starch forms helical secondary structures. Okay, amylopectin helical. Then the linear chain, they are forming some helical structure in case of the starch amylose and amylopectin. Okay, so they forming a complex structure. So this is called the secondary structure. Okay. The linear was the primary structure, and then from the helical is the secondary structure, and then as we find in case of amylose and amylopectin. Okay. So starch can hold I2 molecules. Okay, in the helical portion, starch I2 and forms blue colored complex. So I2, iodine. Okay, starch can hold iodine molecules in the helical portion. Okay, starch and iodine together form a blue colored complex. So cellulose does not contain complex helices. Okay, and hence cannot hold I2. But in case of cellulose, we cannot test in the lab with this iodine. Okay, because in the case of cellulose there is no helical, okay, and cannot hold this iodine. So this starch with the help of this iodine, we can test the starch as the carbohydrate is not. We can test whether it is starch or not with the help of iodine, because it is helical and holds this iodine and it forms a blue colored complex. Okay, so cotton fiber is cellulose. Starch is found in potato, rice. Okay, cellulose is found in the plant, in leaves, in the stems. Okay, plant cell walls are made of cellulose. This you have already studied in previous classes. Paper made from plant pulp is cellulose. That is the cellulose paper. Okay, pulp. There are more complex polysaccharides are needed. So there's so many variety of polysaccharides are there, and then very complex polysaccharides are also found in nature. There's very complex polysaccharides also found. Okay, so what are modified sugar? Derivatives of sugars are called modified sugars or sugar derivatives. Okay, because these sugars belong to carbohydrate and their derivatives are called sugar derivatives. Amino sugars are called are chemically modified sugars where amino group is joined. Glucosamine N acetyl galactosamine. These are the examples of the amino sugars, where amines are attached, amino groups attached, and they are chemically modified sugars. Okay, so here you can see this. Gram-positive bacterial cell wall have a kind of a structure where there is a little difference in the membrane. You can see, is it? Peptidoglycan, then the lipoticoic acid is present. Okay, and in case of this, the outer one, peptidoglycan is present in case of the gram-negative bacterial cell wall. In case of gram-positive, there is the peptidoglycan. Then you can see the lipoticoic acid. Okay, so this is how there is a difference. Cell wall fungi or exoskeleton arthropods made of chitin. This is also a kind of polysaccharide. Okay, here also is a polysaccharide. I forgot to mention that this is the peptidoglycan. This is also polysaccharide. This is also a polysaccharide. So peptidoglycan, which is present in the cell wall of the bacteria, is a polysaccharide, and the chitin is also one of the complex or specialized, or you can say the modified or complex polysaccharide chitin. Okay, this chitin is found in the cell wall of fungi and the exoskeleton of the arthropods. So chitinous plate is present in it on the head region of the upper surface of the arthropod cockroach. That is the chitin, made of the chitin. Okay, homopolysaccharide made with N-acetyl glucosamine subunits. Okay, this chitin is made up of the this compound called, this is a homopolysaccharide and made of this N-acetyl glucosamine subunit. Chitin is made up of the N-acetyl glucosamine subunits. Okay, so that is the chitin. Majority of complex polysaccharides are heteropolymers. Obviously, if it will be the complex, then definitely there will be heteropolymer. The shells of snails, okay, that is made of the heteropolymers. Proteins are linear chains of amino acid linked by hydrogen bonds, no. Glycosidic bond, no. Peptide bond, yes. Ionic bond, no. So correct answer for this question is the peptide bond. This is a kind of amide bond. Okay. So that leads to the, when it leads to the formation of peptide bond, there is elimination of one water molecule, as I have shown in the picture. Okay. GLUT4 is a protein which can act as a hormone, no. Can act as an enzyme, no. Act as an enzyme, no. Can enable glucose transport into cells, yes. They enable the glucose transport into the cells. Okay, that after they can act as intracellular ground substances, no. That's the wrong answer. The correct answer is the number 3. That is the enables glucose transport into cells. Okay. Polysaccharides are long chains of lipids, amino acids, nucleotides, or sugars. It is a polysaccharides are long chains of sugars. Okay, right? Glucose, fructose, etc. It is a long chain. Polysaccharides are long chains of sugars, and sugar being the monomeric unit. Polymer of fructose is insulin, inulin, both 1 and 2, or chain of amino acid. It is inulin. Okay, the polymer of fructose is also a kind of homopolymer, and this is the inulin, and this is the we call the inulin, not insulin. Okay. Now we come to the nucleic acid and structure of protein. Now this is the important part and important for the exam also. Like I told you in the beginning of the class, some macromolecules, like when it's formed a polypeptide chain by when amino acid joins by peptide bond, okay, and these chains are again in turn they are forming a complex structure, okay, and based on the structure complexity, proteins are of different types, like primary structure, secondary structure, tertiary and quaternary, that we'll see when we'll study about the structure of protein. Before that, we'll go through the nucleic acid. Okay, and nucleic acid are other type of macromolecules that were present in the acid insoluble fraction of living tissues. So when you study in the molecular basis of inheritance, when you go to class 12, then you study about the structure of the DNA and nucleotides, nucleosides. Okay, but here we will go through some important points as per our syllabus, and important points that is important for the NEET examination. Nucleic acids are other type of macromolecules that are present in the acid interaction. When there is the fraction of living tissue, okay, present in the acid insoluble fraction, we find that this nucleic acid is one of the important macromolecule, okay, present in the acid insoluble fraction, okay. When we do some research, okay, and we do the acid insoluble fraction of the living tissue cells, then you find this nucleic acid one of the component, one of the macromolecule that is present in the cell. Okay, these are the polynucleotide, okay, formed of the nucleotides, okay, and together with the polysaccharide and polypeptides, this comprises a true macromolecular fraction of any living tissues or cell. So along with this, along with the polysaccharides and polypeptides, they are forming the true macromolecule and found in the living tissue or cells. Nucleotides are the building blocks of the nucleic acid, monomeриc unit of the nucleic acid. So these nucleic acids join together to form these nucleotides join together to form a nucleic acid, like we have seen and observed in case of the deoxyribonucleic acid DNA. So the monomeric unit of the nucleic acid is the nucleotide, okay, that we have seen in the. So here in case of the nucleotide, if you observe the structure of the nucleotide, and we find that there is a phosphate group, there is a sugar group, and there is one nitrogenous base, okay. So these nitrogenous base are different in different molecules. So here nucleotide is the three components are there, and there is a phosphate group, a sugar, moiety, and there is a nitrogenous base. Okay. Distinct components of nucleotides are the three major distinct, as I told you just now. That is the heterocyclic compound. Okay, no, that's sorry. Three major components of nucleotides: heterocyclic compound, monocyclic compound, and phosphoric acid or phosphoric. Yes, the three components. The heterocyclic compound. Heterocyclic compound. Okay, monosaccharide compound, sugar, and the phosphoric or phosphoric acid or phosphate. Okay, so heterosaccharide compound means the nucleic acid or the nitrogenous base named adenine guanine. The one that is so here we can going back to this. So this is the, like I showed you, the heterocyclic compound. Here you can see the heterocyclic compound. Heterocyclic compound, right? Heterocyclic compound. One you saw this. Now another one is the monosaccharide compound, and here you can see the monosaccharide compound, sugar, monosaccharide compound. Okay, so now another one is the phosphoric acid or phosphate. So here you can see the phosphoric acid or phosphate is present here. So this is the, you can see. So this one is the about the three components that I was talking about. Distinct components of nucleotide. Heterocyclic compound, that is the nitrogenous base, monosaccharide compound, sugar, and phosphoric acid or phosphate. So the heterocyclic compound in the nucleic acid are the nitrogenous base named as adenine, guanine, uracil, cytosine, and thymine. So these are the different nitrogenous base present in the as the heterocyclic compound. Substituted by substituted purines. Okay, the adenine and the guanine are the purines. Okay, and there are two types of heterocyclic compound, or you can say the, in case of the nitrogenous base, two group and two types, you can say that purines and pyrimidines. Adenine and guanine are the purines, okay, and uracil, cytosine, thymine are the pyrimidine. Now you can see here pyrimidine. So what is the difference between, you can see the structure right here. So we have got a two cyclic structure, right? And here we have got the in case of this only one ring, only one benzene ring, right? Aromatic ring is present. So these are the pyrimidine. Okay, cytosine, thymine, and uracil. These are the pyrimidine, right? So this is heterocyclic compound. Now about the monosaccharide compound. The sugar found in the polynucleotide is either ribose, a monosaccharide pentose, or deoxyribose. Okay. So if it is there as the sugar is present in two deoxyribose, it is the DNA. If it is the ribose, sugar is present, then it is the RNA. Okay, but if the sugar is monosaccharide pentose. Okay, this is a monosaccharide pentose sugar because there are five carbon atom. Okay, pentose sugar. Okay, so ribose or 2-deoxyribose. Okay, a nucleic acid containing deoxyribose is called deoxyribonucleic acid, that is DNA, and while the ribose is called the ribonucleic acid. One with the ribose, okay, sugar, that is called the RNA ribonucleic acid. Okay, so phosphoric acid or phosphate group is present, and this is here how you can look. This one is the phosphate group, as I told you earlier. Also, this one is the phosphate group. This one is the sugar group, and this one is the nitrogenous base or hydrocyclic compound. This is purine. This is purine, not pyrimidine. Okay, in case of pyrimidine, there is the one benzene. So pyrimidine purine. Okay, so the structure of protein. Now after this, we'll study about this. Now this part is important. Now the previous one also, this is also important, pyrimidine and purine. Okay, but here it's more important, and so many questions are asked based on the structure of protein, and also when you go to higher classes, okay, and you get an entry into your MBBS course, medicine, then you have to study. This concept will be required, and it will be very, very important topic as you enter into the medical colleges. Okay, so proteins are heteropolymers containing strings of amino acids. Like I told you earlier, right? Okay, when the amino acids are joined by peptide bond, they are forming a linear chain, and then these linear chains are joined together to form a complex molecule or macromolecule. Okay, or complex macromolecule. Okay, and this is the complex protein. So it is a simple linear chain formed with the help of some amino acids joined by peptide bond. And now you can observe one kind of bonding interaction between the chain amino acid here, that is the sulfide bond, or it's called sulfide bridge. It is called the sulfide bridge or sulfide bond. This sulfide bond then joining the chains together, forming a complex structure. Amino acids are there. Okay, the structure of molecules explained in different contexts. Okay, in organic chemistry, the structure invariably refers to molecular formulae, example NaCl, NH. Okay, so the structure of molecule explained in different contexts means in inorganic chemistry, when we study about the structure, that means like we are studying about the sodium chloride and magnesium chloride, we are studying about the ionic bonding and all. Okay, so here you can see how ionic bond is formed. Okay, the one, there is a kind of, like we know the sodium is the Na11. So what will happen? The 2, 8 and 3. So what will happen? It will, 2, 10, 1. So 1, 2 at 1. So if it will lose one electron, then it will form, it will be stable. It will acquire a stable configuration, right? And if there will be addition of one electron in case of the chlorine, because it has got 2, 8, 7, right? After 15, this is 7. Okay, so if you get one electron, so from this here, this electron will come here, and this join here, come and plus 1, then it becomes a stable. So it will become 2, 8, 8, and losing this one electron, it will become 2, 8 and stable. So then there is a slight positive charge on sodium atom and slight negative charge on the chlorine atom. So these negative positive charges, they are coming forming an electrostatic bond, ionic bond. Okay. Similarly, in case of the calcium, this is the magnesium chloride. Okay. So two atoms of chlorine atom. Okay, and the one atom of magnesium chloride, they are forming an ionic bond. Okay, like we have seen in case of the ionic bond, or forming the by the electrostatic force of the positive charge of the sodium atom and the negative charge of the chlorine atom. Okay, so that is how this ionic bond is formed. Now, in case of the organic chemistry, and what we do and how we study is the organic chemist always writes a two-dimensional view of a molecule while representing the structure of the molecules, like example benzene, naphthalene, etc. So we write the two-dimensional like here. Okay, because we are concerned with the significance. Okay, while we are writing the molecules, we have to write in such a way, because how we are going to study about this molecule and what is the significance of writing these because we are studying about the living tissues and the chemical compounds that is present in the living organisms. So accordingly, we need to write it. In case of the inorganic chemistry, as I told you, in the ionic, the bond that is formed in the sodium chloride, right? Now, in case of physicists, those who are studying about the physics, they are writing these in the three-dimensional views of the molecular structure. Okay, we are in case of the biologists, as I told you, or organic chemists. Okay, so they are depicting the structure or molecules. Okay, a molecular structure like this. Okay, because we are concerned with the features, or you can say the how this bond and how this structure is related to the functions of the biomolecules. Okay, and in case of this, in case of physicists, they are concerned with the physical structure. Okay, there is the three-dimensional structure or three-dimensional view of the molecular structure. So there are different views in case of the inorganic chemists, organic chemists, and the physicists. Okay, in case of biologists, now we are again. So I told you that there are organic chemists, no, but that's not. But in case of the biologists, here describe the protein structure at 4 levels. Okay, so we are in case of the biology, we are describing our protein structure at 4 levels. Why? Because depending on the different 4 levels, we are able to understand the clear functioning of these different protein molecules and its role in the human body or other animals or other living organisms. Okay, so primary structures, I told you, primary, secondary, tertiary, and the quaternary. So in case of the primary, like we have already seen, you have observed it, a linear chain of amino acids forming a peptide, what we call that polypeptide chain. Okay, the sequence of arrangement of amino acids, that is positional information in a protein, which is the first amino acid and which is the second, and so on. So that is the primary structure. Simple or simply a chain where we know that the different positions of the amino acids are joined end to end by the peptide bond. So this is a simple structure, and that is the called the polypeptide chain, called primary structure. When these are joined, the protein imagined as a line. Okay, the left end represented by the first amino acid, and the right end represented by the last amino acid. Then they are joined. Okay, and what will happen? Okay, then they form this coiling structure. Then they are forming the helical structure. Primary structure is the one which is this is the primary structure, linear chain. This is a linear chain you saw, and this is called the secondary structure. There are two kinds of secondary structures. Okay, beta pleated sheet or alpha helix. So this is the alpha helix, and this is the beta pleated sheet. So this is the beta pleated sheet. Okay, and there is the alpha helix that is present in the keratin. Okay, alpha helix in the hairs. Protein. Keratin protein that is present in the hairs and in the nails. Okay, and that is made of the alpha helix. So that is the coiling. Okay, and they are spirally arranged with the help of the bonding between the spirally arranged polypeptide chain. Okay, there is a kind of a disulfide bonding. Okay, that is called the your alpha helix, or that comes under the secondary structure. So there are two types under the secondary structure. That is the alpha helix and the beta sheet. Okay, which is like a plain sheet. Okay. Now in case of the tertiary structure, you can see this alpha helix is even more complicated. Then again they are forming some kind of globular structure. They are further going some conformational changes, or some kind of foldings and the conformational, and then there is a bonding between the different chains and forming a globular structure, that is the tertiary structure. Okay, so that maybe the albumin, I think the albumin is another kind of protein that depicted as the tertiary structure. And now the quaternary structure, these tertiary structures are again further. Okay, they are forming a complex structure by joining with some other type of bond, hydrogen bonding and disulfide bonding, and forming a more concised, more compact and more complex structure as they find in the hemoglobin. So that is the example of the quaternary structure. Okay, so that is now the first amino acid is called the N-terminal amino acid, and you know that the last amino acid is called the C-terminal amino acid. Okay, so C-terminal amino acid here. C-terminal and the N-terminal amino acids are present in a polypeptide chain. Okay, the thread is folded into form a helical form, similar to a revolving staircase in case of the alpha helix, and a protein thread does not exist throughout as an extended rigid rod. Now, See, only some proteins of the portion of the protein thread are arranged in the form of helix and some are not. Okay? So primary structure is linear, in the secondary structure folding and then some are not and then again it forms. So in case of tertiary structure, you can see this is the tertiary structure, okay, and the quaternary structure forming of assembly of individual protein forming a big complex, a big macromolecules, okay? Forming of, there is already, forming on globular protein and these four globular proteins again forming a more complex, okay, then forming, this is called the alpha helix you can see, and now here in the proteins only right-handed helix are observed, right-handed, not left-handed, okay, so this is how right-handed hand, this kind of, the helix are found in the protein, okay. The secondary structure, protein threads are folded into other forms called secondary structure. They are now, in case of this, the tertiary structure, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to a thready structure, tertiary structure. I told you that is globular protein. So the folding of the chains, then again they are forming the complex, okay, they are again folding itself upon itself and a hollow ball like a structure giving rise to a 3D structure, okay. So tertiary structure is absolutely necessary for many biological activities of proteins. Globular proteins are present in our body that is representative of tertiary structure. And some proteins are an assembly of more than one polypeptides or subunits, okay. So here, the manner in which these individuals folded polypeptide or subunit arrangements in the protein give quaternary structure of the protein. As I told you earlier, manner in which the individual folded polypeptide subunits, like in tertiary structure, then again these subunits forming as a subunit, okay, or as a single unit, but coming to form as in quaternary structure, they are acting as a subunit. The fourth subunit forming a complex macromolecule, then that will be the quaternary structure. Example, linear strings of stairs arranged one upon each other in the form of a cube or plate, etc. Okay, so quaternary structure is the architecture of protein molecules, and here we find that the quaternary structure of adult human hemoglobin consists of 4 subunits. Like I told you, this is example of the quaternary structure. Yeah. So there is a important atom that is the iron, and this hemoglobin is present in the RBC or erythrocytes, which is responsible for carrying the oxygen molecules. Okay, alpha chain, then is the beta chain, okay, then the hem group, okay, and then you can see the alpha chain, beta chain, and the hem group, and the iron is there. So these are from alpha chain, okay, so alpha chain, beta chain, you can see the 2 alpha chain, okay, so this is the beta chain and this is the alpha chain. This is also the alpha chain and this is the beta chain. This is the beta and this is the alpha. Here is written alpha and beta. So these are the 2 alpha chain and 2 beta chains are present, forming a complex structure. So 2 of these are identical to each other, 2 alpha and 2 beta. Hence, 2 subunits of alpha and 2 subunits of beta attack the beta type. They form together a hemoglobin molecule. Okay. Quickly we'll go through some questions. That is, the building blocks of nucleic acids are the amino acids, or lipids, or sugars, or nucleotides. It is the nucleotides. Okay. Heterocyclic compounds in the nucleic acid is ribose, or deoxyribose, or alanine, or purine. It is the purine. Okay. Now, okay, in nucleic acid it may be the pyrimidine also, not purine. But here in this case, in these options, it is the purine. Okay. In a protein thread, last amino acid called N-terminal amino acids, or B-terminal, C-terminal, or A-terminal. Okay, so it is the C-terminal. Okay, thread last. N-terminal first, and the last is the C-terminal. Now, in 3-dimensional view of protein is called the primary is a tertiary. Okay, tertiary structure because it has that 3 dimensions. It is forming a more complex and forming a 3-dimensional. Our human hemoglobin contains 2 alpha chains and 2 beta chains. It's very easy, 2 and 2. So it will be the number 1 and okay. So this was all about the structure of the nucleic acid and the protein. Okay, and before that we studied about the carbohydrate polysaccharides. Okay, these polysaccharides, nucleic acids, and the protein. Okay, these are the macromolecules, and the structure. Understanding this structure is very, very important in understanding the functions of these molecules and the role, how they play their role in the physiological functions of the animals or plant body. Okay, So this was all about the macromolecules, okay, and tomorrow again we'll meet at the same time with another topic, okay. So till then, goodbye, okay, and see you tomorrow again. So take care, bye.